All right. As Pete said, I've been in Europe the last 8 years. I joined about 6 months ago. And yeah, there my big focus was taking non-GMO approaches to breeding better yeast. So I'm a molecular biologist by background. I'm not a brewer. I got an interest in home brewing. And from there, just sort of made the transition into brewing research. Yeah. So if we're looking at flavour in beer, I guess there's 3 key ingredients, and I hope there's no water chemists in the crowd, but I'd say, you've got yeast, malt, and hops that all produce interesting flavours, and you've got like aldehydes and pyrazines from malt, you've got the fruity esters from yeast and the higher alcohols, and you've got all these citrusy and piney and terpenes and floral things coming from hops as well. And it's the interplay between all these flavours that give beer its unique flavour. You think of how many different styles we have, how much diversity we have in beer, that's all from a different profile picked from these ingredients. And they all introduce their own unique flavours, but what we're getting more and more interested in is the interplay between these ingredients and how they work together. And what I'm particularly interested in at the moment is the interplay between yeast and hops. And so I guess the real question for us is how can we take these 2 ingredients and make them synergize to produce something that's more than the sum of both ingredients individually. And the way we are going to do that, or are doing that with things like omega yeast, is through biotransformation. There's a few different types of biotransformation that yeast can do. The first one is release of bound flavour molecules. So you've got the thiol example up the top where they're trapped. You can release them and have the thiol, but you've also got the terpene glycosides where they're attached to a sugar, and yeast is capable, yeast is capable of releasing those aroma molecules. So that's one type of biotransformation. Another type is actually modification of these aroma compounds. So you've got here in green the example where you're getting esterification of those compounds. So you've got 3SH going to 3SHA there, and it's changing the aroma of that compound as well. And the same thing can happen with the terpenes as well. And one other type of transformation is structural rearrangement of those molecules as well. And here you've got the case of geraniol being rearranged into citronellal and changing that flavour profile as well. So these are all transformations that are possible with yeast. So we're focusing at the moment on release of thiols from their bound precursors for the reasons that Laura talked about earlier. They have interesting aroma profiles, so that's always good, but the real reason is because they're active at such low concentrations, right? That's that part per trillion sort of range. So we can try and achieve real changes in beer with just a small increase in production of these compounds. And if we look at a few examples of hops here, we can see that in blue you've got the glutathione-bound precursors, you've got in green the cysteine-bound ones, and in red the free thiols from these hops. You don't even see red on these scales because there's that much precursor trapped in the hops compared to the free thiols. On the left here we have a few hops from freestyle, and in particular the Motueka stands out. It's just got crazy amounts of precursors. You're up to more than 30,000 nanograms per gram in that, which is more than any I've seen reported in the literature at all. If you zoom in on this down the bottom there, you can see that little red line there. That's the amount of free thiol in that hop. So the amount of precursor that's sitting there trapped and waiting for us to release is just a huge, huge resource that we should try and tap into. And if you're looking at the top here, you've got the structure of that glutathione-bound 3SH, and you can see within that structure is actually the cysteine-bound form. So that's one breakdown step that yeast is capable of doing, and then within the cysteine one you can see there you've got the free thiol as well. So yeast can do both of these breakdown steps, and that's what we'll look at. So whenever we're trying to develop a new yeast, I think we have to take a step back and look at the biology behind it and see what targets we can go after. And this is obviously important for a GMO approach where you're picking something in particular and targeting it, but it's even more important for a non-GMO approach where we really have to understand all the biology and try and tap into every little bit that we can to maximize this because we don't have those same GMO tools. So if we look at a yeast cell, they're a single cell organism but they're a little bit more complicated than bacteria. So they are eukaryotes like we are, and what that means is they have different structures within them that operate the cell and its function. So one of those is the nucleus. That's important for us. That's what holds the genetic material of a cell. So that's all the blueprints of all the proteins that are going to be produced in that cell, all the enzymes that are going to be produced, but also the instructions of how and when those will be expressed. Another important organelle for us to think about is the vacuole. And that's essentially the yeast recycling center. So they'll send lots of products they want to break down in there. And it's for waste disposal, but also sometimes a release and reuse of some of those waste materials. So if we're thinking about the thiols, if you've got cysteine-bound thiols outside the cell, they have to be brought into the cell. And you've got the example here of the GAP1 transporter, the general amino acid transporter. That's important for bringing in these cysteine-bound thiols. Once they're inside the cell, you've got those beta lyase enzymes, so IRC7 being the key one in yeast, that can then transform those thiols into the free form, releasing that cysteine. So these are all key targets for us to think about what we can try and improve on in brewer's yeast. Alongside that, you've got the glutathione-bound thiols. They come in through different transporters. They have additional steps where they have to be pulled into the vacuole. They're broken down through proteases into that cysteine form of the thiol. They can then be fed back out into that same beta lyase pathway. So these are all important steps that we need to look at when we're trying to optimize a yeast strain. And what we see here are all the positive aspects that we want to try and improve on. But the single biggest problem in brewing, much more so than in winemaking, is that the control from the nucleus is blocking this with nitrogen catabolic repression. So what's really happening in yeast is that they're not releasing thiols for the hell of it. They're not just always doing it. What they get from this is actually freeing up some nitrogen, right? So in conditions of starvation or whatever, and they want extra nitrogen, they're going to activate this pathway. But when you're brewing and wort, you've got a huge amount of nitrogen that's free for them to use. They don't want to be wasting their time with these kind of pathways. So they're actually purposefully shutting everything off. So even if you've got a great strain that has an active beta lyase, it's probably not going to be using it at all. And that's our single biggest problem at the moment. So that's what we really need to try and tackle and get our way around that. And that's what the GMO yeast have done. They've just put in a new promoter, so they're bypassing all of that control. We can't do that, but what we can do is look for natural variation within these and try and target that. So one example is URE2, which is a central player in that nitrogen control. We can look for mutations in that deactivate that system and try and exploit those. So these are all the points that we're trying to think about when we're developing a yeast. But how do we actually do it? So you've heard about the GMO approach this morning, and it's great. It does exactly what you want, and is very specific, and it's quick. You can produce these pretty quickly. The big issue with it is regional differences. So New Zealand, we don't use GMOs, and even if we did, there'd probably be a public perception problem that would take time for acceptance to happen. It was a big thing in Europe as well. Like, there's just a big public perception against GMOs. When I moved there, it was really, really against it. By the time I was leaving, they were anticipating a change coming, and all the bigger companies are now starting to think, right, we've got to jump onto this and get ready for when it finally does change. So down the track, I think it probably will, but places like New Zealand right now, we need something else. So what we can do instead is look at natural yeast diversity. So this might mean looking outside of beer. We can look at wine, we can look at bioethanol yeast, different things like that, or go totally different and look for non-conventional yeast of different species. So the benefit to this is it's fast and easy. If you've got the yeast there, you can try them and see if they work. The downside is that you're limited to what exists, plus you need a good collection of these yeast, right? You can't just go out screening and expect to find something quickly. You need thousands of yeast to screen for this kind of stuff. And even if you hear of a good species like, Metricoia or Pichia is supposed to be good at releasing thiols, you think that's one species named. You think of how much diversity there is in all the brewing yeast that you use. There is as much, if not even more, per species, right? So even if you hear of a good species, you need to test thousands of those before you're actually going to find the one that's going to work for you. And the other approach is we can take a breeding strategy into this, where we can look for two parents that have different properties that we want, combine them together, select those, and maybe at the same time remove some of the traits that we don't want. The downside to this is it's really time consuming. So if we could do GMO, we probably would. Instead, we're going to stick with this. So how are we exploring natural diversity at Wild Workshop and Garage Project? You got up here the cool ship at Wild Workshop. So obviously there's lots of spontaneous fermentations there. It's a great source of wild and interesting yeast that we can try and isolate and test for different properties. And you can see up here, this is an electron microscope image I took of yeast a couple of weeks ago that had come out of a spontaneous fermentation from Wild Workshop. So that's 5,000-fold magnification of these things, and we're looking at what was in there, and there's at least a couple of different Brettanomyces species that we pulled out of that. So we're building up a collection of all these things that are coming out of Wild Workshop, but at the same time, we're looking to develop it from other places as well. So we'll do a bit in the lab, and we usually start with a small-scale fermentation. So this is a 50 mil fermentation, and in this case, it's got a fresh hop that came from Freestyle the other week, and we'll grow these, look for signs of fermentation, and if they are fermenting, we'll plate out dilutions, usually with an antibiotic in there to prevent bacterial growth, because I'm focusing on yeast at the moment. Once we've got single cells like that, we'll pick those single cells, see what they're like, do a little bit of characterization based on how they look, pick the interesting ones of those and try and build up a little bit of a collection. Once we've got a collection, we start screening them for different phenotypes. What sugars will they grow on? Can they ferment multitriose? Do they produce off flavors? This sort of stuff. And once we've got enough of those that are interesting, we send them off for DNA sequencing to work out what the species is. And this is quite important when you're cherry picking like this. If you're doing spontaneous beer fermentations, you're pretty safe. The conditions will select safe yeast. When you're going out to the wild and choosing new things, you really got to be careful what they are. So you want to make sure you've got safe yeast to use, and bear in mind that some of these things can produce toxins and things like that, so you want to only use safe species, but also many of them can be killer strains to other yeast as well. So you don't want to bring into the brewery something that's going to wipe out your house beer strain or something like that. So we're pretty careful on that. Further to this, we've got a few academic programs running at the moment. So one I want to kickstart at the moment is, it's more of a biological thing where we're looking at microbiomes of yeast, and we're thinking about these wild ingredients, like say a hop. There's different microbes growing on there naturally. And we're asking this question about domestication of those micro communities. So what happens when you take something from the wild like that, put it into a human-made condition like brewing? How are those wild microbes adapting? And can we replicate that in the lab and make stable communities of more than one organism and see how they play together and will they build each other's traits? And then how can we apply that to brewing? So that's something that's more of a longer-term goal. We're just starting that at the moment. But what we are currently doing at Garage Project with some beers is co-fermentation or sequential fermentation. And you'll see that on some of the cans labeled as double shot aroma boost. And what that really means is within that fermentation, they might have started with an interesting yeast and then added a brewer's yeast on top, or put them together, sometimes more than 2 yeast, and often with phantasm as well to try and build up these precursors and pull out interesting flavors. So that's, I think, an exciting way to build layers of complexity to the beers that we're brewing here. The other side of what I'm doing is using breeding programs to generate superior yeast. And we do a lot of this at the university, which gives us access to high throughput technologies that we couldn't have at the brewery otherwise. Some of them are robotics like this, designed for making large libraries of yeast and then testing them in different conditions. So I'll give you a little primer first on the yeast life cycle and how we can exploit it for breeding better yeast. So brewer's yeast is a budding yeast. What that means is it will have a little bud form off the side that grows into a full daughter cell. They separate, and it's clonal expansion. It's just growing itself by cloning itself. But yeast actually have 2 mating types. So they're sort of like a male and a female, but they're called A's and alphas. And when they're in that state, they're haploid, which means they have 1 copy of their chromosome, so 1 copy of their DNA. They don't really want to exist like this. Yeast's preferred state is always to mate together. And they do this in a process called schmooing, where they have a pheromone system where they'll release pheromones, and then they'll actually reach out a little projection to each other, touch their little projections, and they'll fuse into a single cell. And that single cell then is a diploid in wild yeast. So they've got 2 copies of their chromosomes, 2 copies of their DNA, 1 from each of the parents. And that's how we exist as well. We are diploids. We have 1 copy of each chromosome from each of our parents. So any wild yeast you find, and most brewer's yeast should be at this sort of stage. It's where brewer's yeast gets pretty complicated because it's been domesticated for such a long time now. They actually have really messed up genomes. So instead of having 2 copies of the chromosomes like they should do, they have on average 4. And within that, it's pretty fragmented and broken up. So some parts might have 5, some parts might have 3, some might have 2. but on average it's around 4. They're actually likened a lot to cancer genomes because they're so messed up from the domestication process, and some people are actually using them for that kind of research to see how unstable, fragmented genomes develop and what you can do with them to infer onto other aspects of biology like cancer research. But for us, this is where you'll probably find yeast. And there's one more step to the life cycle that is where we really exploit it for breeding, and that's sporulation. So when yeast is put into a stress condition where there's no nutrients, to survive, what they'll do is undergo meiotic recombination. So this is where they're shuffling their chromosomes. This is the actual sexual reproduction part of it. And they'll split themselves into 4 daughter spores, 2 A's, 2 alphas that are then haploids again. And they'll put those in a protective coating, and that can withstand all kinds of conditions. It's really robust. It'll survive for a long time. And then when it's got nutrients again, it'll germinate and it'll go back to A's and alphas and immediately mate with each other as well to form diploids again. That's where we're exploiting it. So what we're doing is breaking open that protective sac, we'll pull out those spores, and then we're controlling who's mating with who. The issue with beer yeast in particular is because they have that crazy genome where you've got at least 4 copies, maybe more of parts. How do you divide that nicely into 4 spores? And the answer is most of the time you can't. They're really, really bad at sporulation. So we have to look for strains that are suitable for breeding, that are capable of sporulating. And even if they can sporulate, they need to be able to germinate healthy spores from that. So it really limits the total number of beer yeast we can use. But wild yeast are pretty good, and there is enough beer yeast to use as genetic stock. So our general strategy is to take 2 parental strains that we've found something interesting in. We'll generate thousands of new hybrids from that through that mating procedure. We'll then try and screen them down on phenotypes like growth, ethanol tolerance, nitrogen requirements, something like that. When we've narrowed it down a little bit, we'll go to lab scale testing, and then we can look at fermentation kinetics and aroma. We'll pick out the best that we like, and we may add a step of back crossing here. So that's, if you think about it, all these progeny are 50-50 from each parent. So if we had picked a brewing strain and a wine strain, it's going to be 50-50 beer and wine. We may want to reinforce those brewing properties. So we may mate back against that brewing parent, and then the second round you're going to be 75% beer strain, 25% wine. And you could do multiple rounds like this. You could make a strain that's 99% a beer yeast, plus you've pulled out those few interesting traits you're after from a wine yeast, for example. And when you've done enough of this and you're happy with the strain, you can move on to pilot scale production and see how it actually performs at a larger scale. So we do lots of this at Victoria University. They've been great to give us space, give me access to their labs at the Yeast Chemical Genetics Lab. So that's actually where I did my PhD, but it was under a different professor. It's now run by Andrew Munkacsky. And so we're collaborating with them, and we're also looking for students. I have my first master's student with Garrett's project starting next trimester, but we're recruiting more, so if you know anyone that's interested, send them our way. But they have some cool technologies there that we use in these processes. The first one's pretty simple in terms of lab things. It's just having access to PCR. So when we're doing these hybridization events, we actually use DNA fingerprinting to confirm that we've got hybrids. So it's the same as paternal testing. We'll have a set of unique primers that will amplify different regions of the genome. They all have different sizes of DNA that it'll produce. We can run this on a gel and see the banding pattern that it produces. And you'll see there that the left 2 ones are 2 different parents, and they have a unique banding signature. And then the hybrids are going to be combinations of that, so we can confirm it's actually a hybrid of these 2 parents. And you might be able to find markers that are useful for tracing. If one of these does really well, we can look for other hybrids that have similar bands in there as well. So that's one thing that we use in the lab. I think the real important thing is the high throughput technologies. So this is what I did a lot in my PhD, and it's what I took to Europe for their development things, was to step up that high throughput phenotyping step. So if we're generating all these yeast, the power of finding something through breeding is really by producing thousands of different progeny from those yeast and testing for rare events that are producing something exceptional. And you can't really do it if you're only producing 4 or 5 strains. You need to go to thousands to look for something exceptional. And we use this robot here a lot, which is a colony replicating robot. So it's designed to make agar plates that you can put yeast colonies on, each one a different strain, up to 1,500 strains per plate. and we'll have whole collections of these plates. And it can make copies of these that we then put into different conditions. So we'll look at ethanol tolerance, nitrogen requirement, temperature tolerance, growth on different stress conditions or media. And we'll take images of them like that after growing them, and you're looking at the size of those colonies as a proxy for growth. So we can do that for measurements like that. We can also include things like, this here is big agar, which has bismuth in it that reacts to hydrogen sulfide. So you get dark colonies if they're high producers of H2S, white colonies if they're not producing much. So we can use that to test for off flavors and say, okay, we only want to use the best ones from that. There was I was talking to someone earlier about killer phenotypes in yeast. So wine yeast especially have these killer traits where they'll actually kill off the other yeast growing there. So they're quite dangerous to bring into a brewery if you end up killing off your house strain or whatever you normally use. And what we can do here is we spot test a strain, say your brewery yeast, and pin new hybrids on top of it, and we can look for each one has that little halo of no growth around it, and that's showing that's got a killer activity to that other yeast, so we wouldn't want to use those ones. And lastly, we can pair it up with a liquid handling robot as well, where we can do micro-fermentation. So in each well of this, we'll have 100 or 200 microliters. We can use brewers wort or different lab media to grow the yeast, and then we can start testing things like phenolic off flavors and measure them in a plate reader rather than having to sniff every single one of them. So you can really go through a massive amount of throughput in these, test thousands of yeast for hundreds of conditions within a couple of weeks. So we use that a lot. And the other cool piece of kit that we have there is a high throughput microscope. So it's actually my PhD, and what I took to Germany first was my specialty of combining all these high throughput technologies with microscopy, and then the image analysis of that and how to look at even more detailed traits, what's happening within a cell. Sometimes growth is a nice phenotype and it's basic, but what's happening within the cell. And we can use this for, here you've got fluorescent tags put on the cells there. So the red we're just using as a marker, and the green one here is a protein of interest that we've tagged, and we're looking at the expression change over time when we've treated them with something. So that's really cool. We can also use it as a normal light microscope. So in terms of hybridization programs, we can use it to test for sporulation in high throughput, how many strains are sporulating, what's the efficiency, and we can use it to test for viability of a lot of strains at once, the same way you would do in the brewery with methylene violet staining and things like that. So that's sort of where we're at. All these projects are just kick-starting now. We're recruiting for students to work on these as well. And I just want to finish on one last thought on what we're trying to achieve with this. So we know these different flavors are coming from yeast and hops, and there's clearly this interplay between the two. And we don't have access to technologies like the GMO yeast, where you can really just say, with precision, this is what I want, I'm going to boost that. What we're looking at instead is to develop yeast that can tweak this flavor balance between them and maybe push it one direction a little bit more to add layers of complexity to beer and make interesting beers overall. So we're not necessarily looking to hit them with a sledgehammer, we just want to shift them into something that's really nice to drink. And yeah, that's where we're at. So I'd just like to thank everyone. This is obviously part of the Hāpi research. We work pretty closely with Freestyle, and we're part funded by MPI and Victoria University for giving us the space to do all this kind of work. Thanks.
Any questions for Peter?
Yeah.
You mentioned earlier in your presentation that these metabolic pathways can be used to unlock phenols, but you're saying most brewing yeast is not capable of doing that. Is that correct?
Most brewing yeast. The thiols you're talking about?
Yeah.
Yeah, the most are capable to some degree. You often see yeast producers saying, oh, we've got full-length ISE, it'll be good for biotransformation. But having a full-length gene is not enough. There's actually a lot of mutations within that inhibit that. So it's pretty rare that you'll find a full-length one with no mutations that's going to be fully functional. And then even if they are capable of doing all those reactions, they're probably not going to do it in brewers' work. There's just too much nitrogen around it, and it's just shutting the whole system off. So I guess that's one thing, is we can try and breed better yeast to get around that system or use a GMO approach. I guess the other thing we could do is just totally rethink how we're brewing, right? Do we need to brew in normal wort all the time? Or can we do something like have a low nitrogen wort and do 2 brews that we blend together to boost these kind of flavors? That would be the other way around. So they're capable of it in certain situations, but probably not in your normal brewer's wort that you're normally doing in a brewery.
So you would say like it's somewhat of a myth with like brewer's wort, or is it particularly thiols or different?
For lots of them, but thiols particularly suffer from this problem. The other one, like the release of terpene glycosides, that doesn't have the same inhibition as well. The trouble there is that those molecules have a much higher flavor detection limit, right? So you need a much bigger impact, or you've got to produce a lot more before you've got that flavor difference. They do work. I think they will make some difference. And even with thiols, there is a small difference there, but it's not going to be that big whack of, oh man, there's heaps of thiols here. But it can be the difference between drinking a beer and thinking, oh, I really enjoy that, and not knowing why. And I think that's a critical thing. Lots of these molecules, like all of them, esters, the terpenes, the thiols, all that, you can have multiple different compounds below sensory threshold, and that combination then has some sensorial impact. And so that can be enough to produce an interesting beer, right? You don't need to be told, oh, this has a huge amount of 3-MH or whatever in it, right? If it's just more interesting from some sort of level. So brewer's yeast, I think, is capable of this. If you select the right strain, you're going to add that layer of complexity to it, but you're not going to get that extreme event that a GMO yeast will produce for you.
Is there work being done to basically isolate the enzyme from the yeast?
There is. Yeah, so for the terpene glycosides, for example, you can get enzymes purified to do that, and it's kind of interesting. I think they're cool. We're going to start playing around with those as well. It's something you've probably got to be careful with how you use them. They're often dirty preparations, so meaning they've purified that enzyme, but there's lots of other activities as well. So you really got to be careful and think about when you're adding these enzymes. You can't just throw them in and think it's going to do something good. So I've seen cases where people try and add one of those enzymes and you add it at the wrong point and you end up with less than what you'd expect, Especially if you release too many of these aroma compounds early in fermentation, you're also scrubbing them out with all the CO2 that's produced from fermentation, right? So what you really want is like more produced at the end that you're going to retain. For thiols, I'm not sure. I keep seeing more and more talk about products that will release thiols, but it seems like it's more of a side activity that they've realized they have some small fraction of it. I've not seen one that I'm convinced is really a good product that really, really boosts it.
Any more questions? No, awesome.